Hello everyone, today we're going to talk about muscle physiology. Quick review of the anatomy of our skeletal muscles. We do have a muscle that's skeletal muscles that are attached to our bones via our tendons. These muscles are made out of units known as fascicles. Each fascicle is made out of multiple muscle fibers. So you can see here's the fascicle right here, and you can see that it's made out of several muscle fibers. Each muscle fiber is made out of several units known as myofibrils, and then you can see them right here, those myofibrils. And then if we zoom in on the myofibrils, those are going to be made out of myofilaments, which are basically the actin and myosin filaments. If we take a look at the muscle fiber and zoom in on it, here's the muscle fiber. You can see that the muscle fiber is protected by a membrane known as the endomysium, and then underneath the endomysium there is the sarcolemma, which acts as the plasma membrane, and the muscle fiber is known as our muscle cells. If we take a look at our muscle fiber and unwrap the sarcolemma and expose the myofibril, there's going to be a membrane right here that you see colored in blue, and that is the sarcoplasmic reticulum, or SR for short. The sarcoplasmic reticulum is extremely important because this is where the calcium ions are going to be stored, and the calcium ions are needed to allow a muscle contraction to go ahead and take place, and we're going to talk about how. And so therefore, the more abundant the sarcoplasmic reticulum is within a muscle, then that would mean the more calcium is going to be stored, and therefore the faster the muscle is going to contract. So if you compare skeletal muscles in general, compare it to your smooth muscles, you will notice that skeletal muscles have the ability to contract way faster compared to smooth muscles, and that's because skeletal muscles have way more abundant sarcoplasmic reticulum compared to smooth muscles. Now, for a skeletal muscle to contract, first thing is you need to have a neuron on top of the muscle fiber, and this neuron right here, when it's right above the muscle fiber, this connection right here, we call it the neuromuscular junction. And this is what we actually refer to as the motor unit. So the motor unit is a bunch of muscle fibers that are innervated by a single motor neuron. Now, what happens is for a muscle to contract, we need to have an action potential or an electrical stimulus that's going to be going down our neuron, and those electrical action potentials are going to make their way down those T-tubules, which are right in between the sarcoplasmic reticulum. As the electrical signal goes down these T-tubules, that's going to trigger the sarcoplasmic reticulum to release the calcium. Once calcium is released, it's going to go ahead and initiate what's known as the cross-bridge cycle. If we zoom in on a myofibril, we would see that a myofibril is made out of myofilaments. Those myofilaments are basically protein filaments that are basically important to allow a muscle to contract. And so those myofilaments, we've got two of them. We've got actin and we've got myosin, and you can see how highly organized they are in the way they are basically set up, and this whole unit is known as a sarcomere. So each myofibril is actually made up of several of those units known as the sarcomeres. Now, as I was saying, when a skeletal muscle needs to contract, an electrical impulse is going to make its way down the T-tubules, and that's when the sarcoplasmic reticulum is going to release calcium. Now, once calcium is released, it's going to go ahead and bind to a protein known as troponin within the actin filaments, and the cross-bridge cycle is initiated, and that's when the myosin heads are going to go ahead and bind to the actin filaments, and the cross-bridge cycle goes ahead and takes place. I do go over the cross-bridge cycle in more detail in the other video, but this is just a quick review so that we could talk about certain concepts that you guys need to know for the lab simulation on Labster. So what I wanted to show you here, when the cross-bridge cycle gets initiated and the myosin heads, they go ahead and they grab onto the actin filaments, you will see that the myosin heads, they go ahead and they basically bend, and that's what we call the power stroke. And when they do that, the actin filaments, you can see the arrows, the actin filaments are going to be pulled towards the M line, and your muscle starts to contract. Now, for your myosin heads to let go of the actin filaments, ATP, which is basically energy, ATP molecule is needed, and it has to bind to the myosin head so that the myosin head can let go. Now, once ATP binds, there is an enzyme actually called myosin ATPase. In myosin, ATPase is going to help hydrolyze or basically break down ATP. And when ATP is broken down, you will get ADP and an inorganic phosphate. Now, why is the hydrolyzation or the breakdown of ATP important? Because once ATP binds and the myosin head lets go of the actin filaments, notice how it's still in that bend position. The myosin heads needs to go back to its original position, needs to go back to what we call the cocked position. And only when it goes back to that cocked position, then it's going to initiate another cross-bridge cycle, and the muscle can contract one more time. And so, therefore, the muscle that's going to contain more of that enzyme, more of that myosin ATPase, is actually going to be able to contract faster. Because as I said, the myosin ATPase is the enzyme that's going to help hydrolyze ATP. And the faster and the more ATP gets hydrolyzed, the more that myosin head goes back to its cocked position, and we can initiate another cross-bridge cycle. And one of the Labster simulations that you guys are going to try out is you are going to be given two different types of muscles, and you would want to figure out which one has more of that enzyme, more of the myosin ATPase. And once you figure out which one has more of the myosin ATPase, then you will be able to predict which one is going to contract faster. And definitely the one that's going to have more of the myosin ATPase is going to be able to contract faster. Skeletal muscle contraction is in need of a steady supply of energy. Some of our muscles are actually different in terms of how they are getting their ATP. So some muscles rely on glycolysis, and glycolysis is known as an anaerobic process. So no oxygen is required, and it takes place within the cytoplasm of the cell. It's a pretty quick process. Not a whole lot of energy is produced from glycolysis. So about a net amount of 2 ATP molecules will be produced, but it's a pretty quick process. So muscles that rely on glycolysis, those muscles are going to be able to actually contract faster. But the thing is, because they are going to be able to contract very fast, those muscles will fatigue very fast. Some muscles, they rely more on aerobic respiration. So aerobic respiration, oxygen is definitely required, and this is the process that happens within the mitochondria of your cells. And so therefore, muscles that are going to rely on aerobic respiration, they are going to have more mitochondria, because this is where the Krebs cycle, or the citric acid cycle, and the electron transport chain takes place. And this is how the muscle relies on getting its energy, by going through these two cycles. Lots of energy is actually going to be released from going through the Krebs cycle and the ETC, but it's definitely going to take longer. So therefore, these muscles that rely on aerobic respiration, they're going to contract much slower compared to the muscle fibers that rely on glycolysis. But because they are much slower, they are actually going to fatigue um, slower compared to the muscles that rely on glycolysis. And so if we take a look at some of these different types of muscle fibers, we refer to them as our fast twitch and our slow twitch muscles. So our fast twitch, those are the ones that are going to be able to contract faster. Compared to the slow twitch muscles, those muscles are going to contract much slower. But as I was saying, they contract much slower, but they are able to resist fatigue better compared to the fast twitch muscles. Those are going to get basically tired very fast. You can see that different parts of your body are going to have different muscle fibers. Different parts of your body might have more of the fast twitch compared to more of the slow twitch. So an example of where you have slow twitch muscles would be within your calf or your gastrocnemius muscle. And those muscles, they are slow twitch, so they contract much slower. They are going to be, if you take a look at them under the microscope and you stain them, they are going to be much darker in color because they have more of the myoglobin. And the myoglobin is a red pigment that has basically high affinity to oxygen. So if you remember, we said that slow twitch muscles, they rely on aerobic respiration, and for aerobic respiration you need lots of oxygen, and so therefore these muscles are going to have more of that red pigment, the myoglobin, and that's why they're going to be much darker in color, and therefore they are actually going to be more fatigue resistant because they are much slower. Compared to the fast twitch muscles, those are going to rely more on glycolysis. So those are more glycolytic fibers. Glycolysis is pretty fast, and so these fibers can definitely contract faster, but they're going to be very easily fatigued. And when you take a look at them under the microscope, they're not going to be have that dark color because they're not going to have as much mitochondria. They're not going to have as much blood vessels as fast twitch muscles. As slow twitch muscles would do. And the reason why they don't need to have as many blood vessels or as many mitochondria, because they don't really rely on aerobic respiration, so therefore they would have less blood vessels, less oxygen, etc. We actually have two types of fast twitch muscles that we are going to talk about right now, but an example of where we have some of our fast twitch muscles would be the muscles that would help you move your eyes. This table right here summarizes the three types of muscle fibers that we have. We've got slow twitch oxidative muscles, or we refer to them as type 1. We have two types of fast twitch muscles. We've got fast twitch oxidative glycolytic, type 2A, and we have fast twitch glycolytic, and that's type 2B. Now, the slow twitch muscles, they are going to be slow because they rely on aerobic respiration. So these are the oxidative muscles because when they go through the aerobic respiration and they go through the citric acid cycle and the ETC, during the ETC, they go ahead and they produce energy via oxidative phosphorylation, and therefore we call them oxidative muscles. And therefore they rely on a high amount of oxygen to be able to go through that aerobic respiration, and therefore the amount of blood vessels is going to be high. The amount of mitochondria that would be within these muscles is also going to be high. And also they're going to look dark red under the microscope because or not even under the microscope, even when you take a look at the muscle itself, you can definitely tell that it's much darker in color, and that's because it has more of the myoglobin, which is that red pigment that has high affinity to oxygen, so it can hold on to more oxygen and therefore help out the muscle. Now we have these muscles in our back to help us maintain our posture, and that's a great location to have them because you don't want your back muscles to get tired as fast. So these muscles are pretty fatigue resistant, and that's our first type, our slow twitch oxidative. Our second type is fast twitch oxidative glycolytic. Oxidative glycolytic meaning that they can go through both the aerobic respiration and they can also go through just the glycolysis. And with more endurance training, you can actually turn that muscle fiber into becoming and relying more on the aerobic respiration. So you can turn them more into becoming fast twitch oxidative and not oxidative glycolytic. So muscle fibers do have some plasticity. With training, you can actually switch one of your muscle fibers into into more of another kind or to act more like another kind. So the fast twitch oxidative glycolytic, as I was saying, with more endurance training, you can make it rely more on the aerobic respiration and therefore make it become more and more fatigue resistant. The amount of capillaries is not as high as the slow twitch, and same thing with the amount of mitochondria, because again, they can also rely on just glycolysis, and so therefore they don't heavily rely on a lot of oxygen. We do find these muscles we rely on them for activities like standing and walking because, again, the muscles in your legs, you don't want them to fatigue as fast. And so, therefore, they are going to be fatigue resistant. Now, they are faster, though, compared to the slow twitch muscles, so they do contract faster, and they contract faster, and one reason is they contract faster because they actually have a higher amount of the myosin ATPase. And if you remember, as I was saying, this is the enzyme that will help hydrolyze ATP for you. And the faster you can hydrolyze ATP, the faster the muscle is going to basically contract. Now, the fastest muscles that we have, the fastest muscle fibers that we have, those are our fast twitch glycolytic. And the reason why they are the fastest is because they rely on glycolysis. And so glycolysis, it's basically, it's more of an anaerobic, respiration. And so therefore, it's much faster. And so the amount of capillaries, the amount of mitochondria, the amount of myoglobin, it's going to be very low, and so therefore they're going to be pale in color. And again, they don't rely on oxygen, and so therefore you're going to have very lower, definitely a lower amount of mitochondria and capillaries within these muscles. And therefore, the myosin ATPase activity is definitely going to be high. They're going to be able to hydrolyze the ATP faster and contract faster, but because they are super fast, they're going to be easily fatigued. And so you will find that you rely on these muscles when you want to, like for example, jump. So again, you want that burst of energy to help you jump really fast, but you will notice that you will get tired pretty fast. And so basically for those like quick, fine movements, you would want to have more of those fast twitch glycolytic fibers. Now it turns out that we actually inherit these fibers, and some of us inherit more of the fast twitch glycolytic fibers. Some of us can actually inherit more of the slow twitch oxidative fibers, and therefore you find individuals that are better in sprinting, and that's because they have more of the fast twitch glycolytic fibers, so they would do really good, you know, in running really fast. You will find individuals that maybe are better in running marathons where they would need to, you know, run for longer distances. They would have more of the slow twitch fibers, and therefore their muscles would fatigue less. So the Labster simulation that you're going to try out, they're actually going to guide you in dissecting two muscles. You're going to take a look at them, and you're going to stain them, and you're going to take a look at their enzyme composition. So you would want to figure out whether they have more of the myosin ATPase or whether they have more of the oxidative enzymes. So you would go ahead and take a look at them, and those muscles, they are going to be different in how fast they are going to contract, or basically how fast they can twitch. A twitch is basically a muscle contraction. The force of their contraction is also going to be different, and basically the time to fatigue is also going to be different for these muscles. The muscles that you are going to take a look at are the soleus muscle and the extensor digitorum longus muscles, which are basically two muscles in the leg. And this right here, they're showing you the muscles in a rat, and those are the two muscles that you are going to be looking at when you are working on that Labster simulation. In the Labster simulation, they're going to ask you to go ahead and stain the muscles, and basically within the simulation, they talk about the technique that you're going to be using, and that is the histochemistry technique. And histochemistry is basically a technique that's used to visualize the chemical properties of the cells and the tissues, and they use special stains to go ahead and see that. They go through three different assays that they talk about in that activity, and the three enzymes that you would want to go ahead and look at or discuss which muscle fiber would have more of it. Myosin ATPase. We already talked about that one, right? We already talked about how this is the enzyme that will help you basically hydrolyze ATP, and this enzyme would help you distinguish between slow and fast muscle fibers. So the muscle fiber that's going to have more of that myosin ATPase would tell you that this muscle fiber is a fast muscle fiber. When you go ahead and you again perform that staining procedure, and at the end look at the results, and you see that this muscle has less of the myosin ATPase, that would tell you that this muscle fiber is a slow muscle fiber. You will also take a look at something known as AGDPH, and that's going to tell you basically the glycolytic potential. So the higher, basically the higher activity would tell you that this muscle relies more on glycolysis. The lower, it would tell you that the muscle relies more on oxidative aerobic respiration. And third enzyme, succinate dehydrogenase, is going to help you distinguish whether the muscle is oxidative or non-oxidative. So oxidative would tell you that those muscle fibers rely on aerobic respiration. Non-oxidative muscle fibers, those are the ones that don't rely on aerobic respiration. And again, definitely the ones that are going to be oxidative, they're going to be much slower muscle fibers compared to the non-oxidative muscle fibers. So that's the first activity. The second activity that you're going to try out with Labster is going to discuss a topic known as muscle twitch. And basically, a muscle twitch is when your muscle contracts, and your muscle contracts after it receives a stimulus. Once it goes ahead and a stimulus initiates your muscle contraction, well, that stimulus is going to first initiate an action potential, which is basically that electrical signal. And after it receives that stimulus and basically an action potential, the muscle is going to go ahead and contract, and eventually it's going to relax. So basically, a twitch is basically a muscle twitch is representing a single muscle contraction, and there's something known as the latent period, and that's basically the time between the stimulus and the contraction of the muscle. So usually the electrical impulses are faster compared to the contraction of a muscle. And the reason why after a stimulus there's kind of like a lag time before the muscle contracts, because during that latent period, the cell is going through basically the electrical impulse is going down the T tubules, the sarcoplasmic reticulum is releasing calcium, the calcium is binding to troponin. So during these steps, the muscle has not contracted yet. And so therefore, you will find a period of time called the latent period, where even though the stimulus has been fired and an electrical impulse has been fired, the muscle doesn't contract immediately. So this is basically showing you the relationship between a muscle twitch and what happens during that latent period right there. Now, another concept that we want to talk about is the length of a muscle sarcomere and the tension. The tension, basically, a muscle tension is basically the force that's going to be applied during that muscle contraction. Basically, how forceful that muscle contraction is. That's basically the definition of a muscle tension. And so for you to have a maximal tension, a maximal tension is going to be achieved when the sarcomeres are at normal length during their resting period. So right here, if we take a look at this sarcomere and you see the actin and the myosin filaments, This is the best length for your actin and for your myosin filaments to achieve basically a pretty high muscle tension. You can see that the muscle tension, basically, again, the force of your muscle contraction starts to decrease. It starts to decrease if you stretch out the muscle. So the more you stretch out your muscle before allowing your muscle to contract. If you go ahead and stretch it out and then go ahead and initiate a muscle contraction, the muscle contraction, the force of that muscle contraction is going to be very, very minimal. Same thing if you go ahead and squeeze the sarcomere, and now you have a whole lot of overlap between the actin and the myosin. That's definitely also going to decrease the muscle tension. So one of the Labster simulation that you're going to do is you're going to go ahead, take the two muscles that you've just dissected, and you're going to go ahead and stretch them, or basically compress them, and take a look at how is that going to affect the muscle tension. So one of the Labster simulation activity that you're going to go ahead and take a look at, you're going to go ahead and stimulate the soleus and the EDL muscles to contract, so you can visualize the muscle twitch. And we do know that when a muscle is stimulated, basically you're going to have that latent period, and that's when, again, the electrical impulse is going down the T tubules and the sarcoplasmic reticulum is releasing the calcium, and then the calcium binds to troponin. So that's all happening during the latent period. And then once the muscle contracts, the cross-bridge cycle goes ahead and takes place. And eventually the muscle will go ahead and relax. For the muscle to relax, ATP is definitely needed so that the myosin heads can let go of the actin filaments, and the calcium ions, they need to be pushed back into the sarcoplasmic reticulum. Because as long as you have calcium around, then more cycles of the cross-bridge cycle can keep taking place. Another activity that you're going to go ahead and take a look at is you're going to take those two muscles that you've dissected, and you're going to increase or decrease their muscle fiber length, and discuss how basically stretching them out or compressing them affects the muscle force, affects the muscle tension. So as I was talking about it in the previous slide, the length of the sarcomere affects the muscle tension. So the muscle tension is definitely directly proportional to the length of the sarcomere, and the more stretched out or compressed, that's definitely going to lower and decrease the muscle tension. Another concept to talk about is the force of contraction. So when a muscle is hit by repeated stimuli, or basically repeated action potentials, if there is time, if there is enough time between those repeated stimuli, the muscle is going to fully relax. But if those repeated stimuli, they're right after each other and there isn't enough time for your muscle to relax, then what's going to happen is that's going to result in basically a more forceful contraction, and that is known as summation. And the reason why it's more forceful, because you're not giving your muscle enough time to relax. So it's not fully relaxed, and it goes ahead and it starts another contraction. So definitely the force of the contraction is going to increase. And as I said, that's known as summation. Tetanus is when you have repeated stimuli or repeated action potentials. And again, your muscle is not fully relaxing and it reaches a maximal contraction. That's basically tetanus. Now, we actually have two types of tetanus. So let's first take a look at single twitches right here. So you can see that here's the, on the x-axis you are graphing time, and then on the y-axis is the muscle tension, or basically the force of the muscle contraction. And you can see that between each twitch, if you a twitch is basically like a muscle contraction. So between each twitch, if you have enough time, then you can see the muscle is fully relaxing before it goes ahead and it starts another twitch. But if you go ahead and you have a muscle twitch right there, and then you are basically starting another muscle twitch. But notice how here the muscle was not able to relax fully, and this muscle was hit by another stimulus, so it didn't fully relax yet, and it started another contraction. Notice how the force basically increased, and that's when you are showing summation, when the stimuli are basically super close to each other and your muscles are not getting enough time to relax fully. Now, that summation can basically lead to either unfused tetanus or complete tetanus, or we call it fused tetanus. So what is unfused tetanus? That's when the stimuli are far enough apart to allow the muscle to relax just a bit between the stimuli. So as you can see right here, the stimuli, it's basically like frequency is really high and the muscle is not fully relaxing. And again, if the frequency of the stimuli increases, you will notice that eventually the muscle tension keeps increasing, but you never reach that fused tetanus because the muscle is still slightly relaxing. And that's what's known as unfused tetanus. And basically that's the maximum tension though it can achieve. It cannot go higher than that. With complete tetanus or basically fused tetanus, that's when, let's say here you have a single twitch, and let's say, okay, enough time has passed and another muscle twitch is about to begin, but you can see that the frequency of the stimuli is extremely high. Notice what's happening. The muscle is not even what? The muscle is not relaxing, and so therefore you reach that maximum tension. But notice how, because the muscle is not relaxing, you have what we call that fused tetanus right there. Now if the muscle basically fatigues, then tension is going to start decreasing rapidly. So as you can see, eventually your muscle is going to fatigue and it's going to lose its tension pretty fast, despite the continuing stimuli. So despite the continuing stimuli, once your muscle is tired, that's it. Its tension is going to drop. Now, there is another concept to discuss, and that is motor units. A motor unit is basically one motor neuron, and that one motor neuron is innervating a bunch of muscle fibers. So a bunch of muscle fibers innervated by one motor neuron is known as a motor unit. So you can see right here this purple neuron innervating those purple muscle fibers. That's one motor unit right here. So in this image, we are looking at 3 motor units. Now, if we take a look at this orange motor neuron, and it's innervating, let's say, those orange muscle fibers. These muscle fibers that it's innervating are all of the same type. So meaning that if the muscle fibers within motor unit 3 are slow twitch fibers, then the whole entire motor unit is a slow twitch fiber unit. If those muscle fibers right here, the one is in blue, those are fast glycolytic fibers. And again, the whole entire motor unit is a fast glycolytic muscle fiber. So each motor unit contains the same muscle fiber types. But as you can see, within one muscle, so if this is your biceps muscle right here, within one muscle, you can have the 3 different muscle fibers that we've talked about. So in one muscle, you can have your slow twitch, your fast oxidative glycolytic, and you can have your fast glycolytic muscle fibers. But one motor unit just has one type of muscle fiber. Now, motor units, basically the number of muscle fibers in a motor unit can vary. So I can have in motor unit number 1, maybe 3 to 5 muscle fibers. I can have in motor unit number 2 more muscle fibers. So the number of muscle fibers within each motor unit can definitely vary. In muscles that are used for fine motor action, for example, moving your eyes, moving your hands, your fingers. So those are like fine movements, or again, like delicate, and you have like more, you use them for fine motor, basically control. These muscles, their muscle fibers, or their motor units contain a very minimal amount of muscle fibers. So these motor units could have like maybe 3 to 5 muscle fibers. But in muscles that are used for more of the gross motor actions like standing and walking, like the muscles basically in your legs, each motor unit in your leg could have about 200 muscle fibers in each motor unit. And as I was saying, basically all the motor fibers in a single motor unit would be the same fiber type. And as you can see, also if you want to recruit, it's called recruitment. If you want to have a stronger muscle contraction, or basically, let's say you want to carry something that's pretty heavy, then you want to go ahead and recruit and activate more motor units. So additional motor units is definitely going to increase your contraction force. One of the Labster activities that you're going to try out is you're going to take the 2 muscles that you've previously dissected, and you're going to go ahead and stimulate them several times so that you can basically take a look at how is that going to affect the muscle tension, and so you can achieve basically summation and tetanus. And you're going to take a look at whether you have basically accomplished a fused tetanus or an unfused tetanus. And you're also going to take a look at the role of calcium in the muscle contraction. So as I was saying, the sarcoplasmic reticulum and calcium stored in the sarcoplasmic reticulum is super important in allowing your muscles to contract. So definitely the more sarcoplasmic reticulum a muscle has, and therefore the more calcium it's storing, the faster the muscle can contract. Another activity you're going to go ahead and take a look at is you're going to investigate the muscle tetanus. And as I was saying, you're going to see whether you've achieved a fused or unfused tetanus. And then another concept to talk about is, why do our muscles fatigue? Well, our muscles could fatigue and maybe because the amount of ATP produced is decreasing. The glycogen, which is our stored sugar in our muscles, it's depleting. Maybe the amount of calcium that we have stored is a very low amount, and so therefore not enough calcium is being released out of your sarcoplasmic reticulum, causing your muscle contractions to actually decrease, and therefore your muscles would fatigue faster. It could be that you have a buildup of lactate, or basically lactic acid, making your muscles more acidic. That can all cause your muscles to fatigue. And it could be that at the very beginning, when your muscle is receiving the signal from the motor neuron, the amount of neurotransmitters or acetylcholine is decreasing, and so therefore you have a decreased action potentials coming down your T-tubules, and therefore that's definitely going to affect your muscle contraction rate. So there is a whole bunch of reasons why your muscles could fatigue. It could also be like a decrease in the motor neuron activation itself that can be also causing your muscles to fatigue. And then, as we said, different muscle fibers fatigue at different rates. So we already discussed how if you're comparing, let's say, your slow twitch muscle fibers and your fast glycolytic muscle fibers, we've already discussed how your fast glycolytic muscle fibers, they're going to fatigue way faster compared to the slow twitch muscles, and that's because the fast ones, even though they contract really fast, because they rely on glycolysis and they are contracting really fast, they have more of the myosin ATPase, they're going to fatigue basically very fast compared to the slow twitch fibers. So another Labster activity that you're going to go ahead and try out, you're going to investigate muscle fatigue for both the soleus muscle and EDL muscles, and you're going to try to figure out which muscle out of the two is basically more fatigue resistant, or it's easily fatigued.